In this final lesson on LTP, we'll look at how LTP can be synapse specific and associative. We'll be using the tag capture model. Now, you'll recall from last time we said that activity at a synapse, if it's a sufficiently high frequency stimulation here, can generate early LTP and then late LTP. But the late LTP requires protein synthesis. It's as if these plasticity related proteins have to then diffuse back out to the relevant synapses to help make long-lasting modifications of the synapse. If there was no activity at a synapse, then that synapse will not enjoy LTP. So the way this would work then is that whichever synapses were recently active would be tagged somehow biochemically. A synapse that was not active would not get that tag. And the idea is only the tagged synapses would be able to capture the plasticity related protein products. And so we have the tag and capture model. And this would account for the specificity of LTP. We'll take a look at that same idea, the specificity of LTP, with a more elaborate diagram showing our data records here as well. So let's say we induce LTP at synapse 1 with high frequency stimulation, and here we see the data showing the LTP, long-lasting potentiation of the synapse. Now, we think that late phase was the result of the fact that the target cell did fire in response, and so proteins got made, and those proteins then can help make long-lasting changes at the synapse. But notice down here at synapse number 2, we're simply just recording the strength of that connection, sending test pulses well before we do LTP here at the synapse number 1. This is the response. After we do the high-frequency stimulation, we get no change at synapse number 2. So synapse number 2 was not strengthened, even though we induced LTP at synapse number 1. So this shows the specificity of LTP. And again, the reason why, according to the tag and capture model, is that these plasticity-related protein products can only be utilized by recently tagged synapses. Having no activity here at this synapse here, they can't make use of the plasticity-related products. So LTP shows specificity. Now, what's the evidence of this tag and capture model involving these plasticity-related proteins? Here's one of the early experiments. So researchers sent high-frequency stimulation down this pathway here at this synapse, inducing early LTP and late LTP. And they didn't do anything here. And we know that there's going to be no LTP at this synapse. In fact, they found no LTP here. They did find LTP here. Now they're going to do something interesting. Within the time window in which these plasticity-related products were being sent out to the dendrites, remember they were triggered by LTP at the first synapse. Within this time window, about an hour, they went ahead and stimulated this second pathway, high-frequency stimulation here. This high-frequency stimulation should be enough to induce LTP, but at the same time they did the stimulation, they blocked further protein synthesis. They wanted to see whether the plasticity-related products that were triggered by the first LTP could be used at the second synapse. So they didn't want any more protein synthesis to happen. They wanted to see whether these already existing proteins could be utilized by the synapse. Sure enough, they got late phase LTP. The suggestion was the second synapse was able to capture the plasticity-related proteins triggered by the activity at the first synapse. Now, the tag and capture model also helps us understand the associativity of LTP. And what we mean by that is the pairing of a strong stimulus and a weak stimulus. Both of those pathways can be strengthened at the same time. We're going to call that an associative property of LTP. Let's consider this scenario. Here we have synapse number 1, and we're going to send high-frequency action potentials down here, sufficient to generate target cell response and induce both early and late phase LTP at synapse 1. At the same time, we have a weak pathway here. We're going to deliver low-frequency stimulation here. Now, that's going to activate the synapse, so the synapse will be active, right? But we know from earlier experiments a low-frequency stimulation will not produce late-phase LTP. Yet at synapse number 2, we record late-phase LTP. So pairing a weak synapse and a strong synapse, so a weak pathway and a strong pathway, pairing them allows us to get late phase LTP in the weak pathway. And the idea is, because both pathways were active, both synapses get tagged. But remember, the high frequency stimulation generated target cell action potentials, so then we get the triggering of plasticity related products, and those plasticity related products then can help both synapses, because both were tagged, so now both synapses can enjoy the late phase modifications due to the activity of those plasticity related proteins. The ability of a weak synapse to be strengthened when activity in the weak pathway is paired with the strong pathway means that LTP can support associative learning. And associative learning is when we associate one stimulus with another. A simple example is classical conditioning. Here we see the dog looking or smelling the food, and the dog begins to salivate. Now, psychologists would say the food is the unconditioned stimulus, and the salivation is the unconditioned response. But now let's come along and just ring the bell. So no food present, so sometime later we simply ring the bell. Now the dog might be interested, but that bell has no significance to the dog at this moment. We would say it's a neutral stimulus, and it's called the conditioned stimulus. Now the idea is, through this training procedure, we're going to try to get the dog to associate the bell with the food by pairing the presentations. So we're going to repeatedly pair bell and food. So we'll ring the bell, present the food, ring the bell, present the food. And so in the dog's brain, the idea is the dog will begin to associate the ringing of the bell with the presentation of food. And so if we do this enough times, at a later time, if we simply ring the bell alone, the dog will salivate. This is called the conditioned response. So previously, the bell was a neutral stimulus. The dog did not salivate. The bell was not associated with food. But when we repeatedly pair the bell with the food, the dog begins to salivate to the bell alone. The dog has associated the bell with the food. At this point, the bell predicts the food. So this is a simple example of associative learning, and we can use LTP to help us understand what's happening biologically to permit this kind of learning. We'll take a simple diagram like this. We have two neural pathways that converge on a target cell. The food pathway, the unconditioned stimulus pathway, initially has a strong connection, right? Multiple synapses there on the target cell. So we would say this is a strong pathway. Presentation of the food by itself is sufficient to drive the target cell to a threshold, and we'll get activity in the target cell, which we're going to call salivation, the unconditioned response. Notice, initially, the way the wiring is, the bell pathway, conditioned stimulus pathway, is weak. So that's a weak synapse there. So activity in the bell pathway is not sufficient to drive the target cell to threshold. So we do not get salivation initially when the dog hears the bell alone. However, as we train bell, food, bell, food, bell, food, we go through the repeated pairing of the bell and the food. Then, after the training, we have a situation like this. The bell pathway has strengthened. We have more synapses here, for example. So now the bell alone is sufficient to drive the target cell to threshold, and we'll get salivation with just the bell. So by repeatedly pairing the bell and the food, we've created conditions to strengthen the weak pathway. You'll recall it's the food pathway that was strong to begin with, and so we will be activating the target cell. But if we are pairing the bell and the food, then the bell pathway is active when the target cell is firing, and that means we have the conditions for LTP to occur at those synapses. And what we're saying here is that actually the strength of the synapse is being increased, and in fact you can actually get the growth of new synapses. So a couple things to notice about simple associative learning, it requires the convergence of neural pathways. If we're going to associate the bell and the food, we have to have some convergence onto a target cell so that we can strengthen those weak synapses. The weak synapses are the ones that are representing the neutral stimulus. Initially, the bell had no sort of significance. It was not associated with food, but after training, the bell is associated with food. And secondly, that synaptic plasticity is the basic biological mechanism for how two stimuli can be associated. Changes to synaptic strengths is what underlies associative learning.